This is the 2022 Online Investor Conference of Microbio Company Limited. We are very pleased to be joined online by the distinguished guests and the investors in this virtual investor conference. First of all, I would like to introduce to you the management team: Chairman Xinsho Kua, Vice President Wanjian Chen, Assistant Manager of the Finance Department Ingchen Wang. Next, I would like to invite Xinsho Kua, the Chairman, to give a brief speech.
Dear shareholders, ladies and gentlemen, good afternoon. Thank you very much for taking the time to participate in the 2022 Online Investor Conference of Microbio Group. We will take this opportunity to explain to all shareholders the important operating situation of Microbio Group this year. As early as 2000, Microbio has laid out a plan for new drugs of symbiotic fermentation of intestinal microecology, and the first product, Chemo-Young Oral Solution MS-20, was approved and launched in 2011 to meet the needs of clinical adjuvant treatment of cancer patients. Based on the clinical experience of Chemo-Young Oral Solution, Microbio continues to inject R&D momentum to study human gut microbiota and to conduct clinical research on intestinal ecology with a number of large hospitals to develop the world's only intestinal microecology drug that can regulate the intestinal microenvironment. This year, microecological new drugs have been applied to the microenvironments outside the intestines, such as the skin, nasal cavity, etc., so as to broaden the application scope and dosage forms and create more possibilities for microecological new drugs. In addition, in order to diversify R&D risks, Microbio Group has laid out a strategy in biotechnology holdings, and we are happy to see that the reinvestment companies can have stronger R&D momentum and fruitful R&D results to create shareholders' equity. Finally, thank you again for being with Microbio, and hope we all together witness more innovative and cutting-edge R&D achievements from Microbio in the field of new drugs.
Next, the Deputy General Manager, Wanjian Chen, will explain the development of Microbio's core undertakings.
Hello everyone, now let me introduce the core business of Microbio Group. The core business of Microbio Group has three main areas. The first is new drug research and development, the second is biotechnology investments, and the third one is organic agriculture, which will be articulated in the following briefing. First of all, the first part is the research and development of Microbio new drugs. Microbio has committed to the development of Symbiota, a new drug of microecology, and MS-20 is the first new drug developed by Microbio. MS-20 Chemo-Young Oral Solution is the first new drug in Taiwan to improve the side effects of cancer chemotherapy and was approved in 2011. At that time, the approved indications were to improve the symptoms of fatigue, loss of appetite and poor social function in cancer chemotherapy patients. At that time, we also found that the new drug targeting the intestinal microecology has the ability of changing bacterial compositions. In the past 4 years, the revenue of Microbio Chemo-Young Oral Solution has also increased year by year, and it has reached 63 million new Taiwan dollar this year. Next, we are actively developing new indications for the Chemo-Young Oral Solution as a strategy to expand the marketability of MS-20 itself. The market for cancer immunotherapy is huge and is considered as an important key treatment for novel cancer therapy. The cancer immunotherapy market was valued at $85 billion in 2021 and is expected to reach $227.1 billion in 2030 based on a CAGR of 12.6%. Among them, the sales of antibody Keytruda, an important anti-PD-1 antibody for immunotherapy, have also increased year by year, and the annual sales value has exceeded $17 billion by 2021. Which is currently the best-selling drug in the world. In recent years, many scientific studies have found that the microbiome of the intestine determines the effects of tumor immunotherapy. What was seen from the 2018 Science journal literature is the composition of cancer gut bacteria, which determines the response rates of drugs for immune checkpoints. The 2017 Cell journal literature also found that gut bacteria can determine the recognition of tumors by the immune system, affecting the efficacy of immunotherapy. The 2019 journal Nature literature also found that the composition of intestinal bacteria determines CD8 T cells, which are key effector T cells for immunotherapy to generate activity, which in turn affects anti-tumor immunity. In the 2018 Gut Journal literature, it was also found that intestinal bacteria can directly influence the chemotaxis function of immune cells by modulating cytokines, promote immune cell infiltration of tumors, and elicit the efficacy of immunotherapy. In other words, research has identified the intestinal microbiome as a key factor to determine tumor immunotherapy. In recent years, the journal literature has more directly discussed that the key to the effective efficacy of anti-PD-1 is actually influenced by several important key gut bacteria, among which another science journal literature reported Akkermansia is one of the important key bacteria affecting PD-1 cancer therapy. In addition, this year's Nature journal literature also found that the diversity of intestinal bacteria and Bifidobacterium longum are important controllers affecting the effectiveness of PD-1 treatment for lung cancer. Our product MS-20 Chemo Young Oral Solution is composed of metabolites of a variety of probiotics, which have the function of regulating the bacterial composition. When we can regulate the key bacteria that affect tumor immunity, we have the opportunity to enhance the efficacy of existing immunotherapy. In recent studies, we have found that the gut microbiome can be reshaped in the people who take MS-20, and the richness and the diversity of their gut bacteria can be increased. For some key bacteria, including those just mentioned, Akkermansia or Bifidobacterium longum, that can improve the efficacy of anti-PD-1 immunotherapy, MS-20 can also significantly enhance their effects. And we have also seen in preclinical mouse trials that when MS-20 and anti-PD-1 drug are combined, the survival rate of mice can be greatly improved. You can see the figure on the left. The red line refers to the effect of anti-PD-1 antibody on mouse survival, and 33.3% of mice have a response to the treatment. The combined use of MS-20 can greatly increase the survival rate of mice from 33.3% to as much as 75%. If the results of such a preclinical model can be clinically verified, which means MS-20 can effectively increase the response rate of current anti-PD-1 therapy, and this will make MS-20 more marketable. At this stage, the clinical trial with the combination therapy of MS-20 and immune checkpoint inhibitors is currently underway, and we have been cooperating with the hospitals in Taiwan to accelerate the indication for the combined use of immunotherapy for non-small cell lung cancer patients and carry out patient admission. In addition to immunotherapy, we are also actively conducting exploratory clinical trials on other indications, including the cachexia in pancreatic cancer patients and inflammatory enteritis to explore whether MS-20 has an effect on treating IBD. These clinical trials are currently in progress. Therefore, for the MS-20 expansion strategy, we mainly have three strategies. The first is to explore new indications that can be applied through exploratory clinical trials, and then we can design phase 3 clinical trials to expand our MS-20 new indications. In addition, we are also evaluating new routes of administration, because MS-20 achieves its efficacy mainly by changing the microbiota, thereby increasing the effects of existing therapies or treatment-related indications. So we are now evaluating whether we can directly use rectal administration in addition to the oral dosage form to apply our products to the location of gut bacteria, and thus more efficiently achieving the effect of changing the bacterial composition. This route of administration is also being evaluated. Also, for the part of the U.S. market, because the first new microecological new drug launched in the United States this year, we also evaluate the use of the U.S. market in view of the fact that Symbiota is a very special product, a symbiotic metabolite mixture of a variety of probiotics. We will first apply our products through the medical food category in the United States, in order to shorten the entry time and expand the MS-20 market in the United States. In addition to the first-generation product being is a metabolite mixture of a variety of intestinal bacteria through anaerobic symbiosis, it has just been mentioned that MS-20 has passed the Taiwan New Drug Certification, and we are now actively developing and expanding new indications. The technology of our microecology platform is actually to regulate specific bacteria by increasing beneficial bacteria and inhibiting pathogenic bacteria to achieve the purpose of symptom improvement or disease treatment. At present, via this technology platform, we have developed 1,200 microecological libraries in the past 2 years, so that we can screen for suitable effects on microbiome in ways of the proliferation and inhibition of different bacteria to develop new drugs. On the basis of the niche of microbiome microecology new drug, in addition to our world-leading patented technology since 20 years ago, we have now also found specific intestinal ecology drugs, which has the ability to increase specific good bacteria, inhibit specific bad bacteria, and change the microenvironment of the intestine or other organs. The key is that no viable bacteria are required, and without live bacteria, colonization is not required to its activity, and it also has a high stability and a good safety. Our concept is to regulate immunity by influencing the bacterial compositions and thus affect the microenvironments of human organs, which can be used alone or in combination with existing drugs to accelerate the development process. In addition to oral dosage forms to improve the intestinal microecology, this year, we also have skin dosage forms, nasal dosage forms or rectal dosage forms to expand the application of microecology products. Next, we will report the development of new drugs for the new generation of microbiome microecology drugs, and we mainly develop 3 major indications, including cancer, autoimmune diseases, and diabetes. First of all, the first microecological new drug product we developed is MB107, a new drug for skin microecology, which has the effect of AHR activator. AHR is regarded as a target for a new generation of drugs, and tapinarof, an AHR active activator developed by Dermavant Sciences this year, is a non-steroidal topical cream treatment, which has completed 2 phase 3 clinical trials of plaque psoriasis, and the efficacy in these phase 3 clinical trials is very significant. In August this year, it passed the FDA's NDA review and is listed in the US. In other words, this target is already proven to be a very critical target. In addition to the indications for plaque psoriasis, Dermavant also launched 2 phase 3 clinical trials for the treatment of atopic dermatitis in September 2021, expanding its indications for atopic dermatitis. The market potential of MB107, similar to tapinarof, is an activator of AHR with the potential to treat plaque psoriasis as well as atopic dermatitis. Plaque psoriasis was marketed at $14.2 billion in the 7 major countries in 2020 and is expected to grow to $27.5 billion by 2030 at a CAGR of 6.8%. Atopic dermatitis reached $12.7 billion in the global market in 2021 and is expected to reach $27.6 billion in 2030. Those are 2 indications with great potential value in the skin market. We have completed a lot of in vitro studies of MB107, and we will introduce it to you later. First of all, MB107 was tested in vitro for direct comparison with tapinarof, in which MB107 exerted its AHR potential and achieved a comparable effect to that of tapinarof in the expression of skin barrier genes. In the preclinical animal studies of 2 indications, psoriasis and atopic dermatitis, MB107 has reduced redness and scaling symptoms, and has fairly close efficacy results compared to tapinarof. This year we completed exploratory clinical trials in psoriasis and atopic dermatitis. In patients with psoriasis, after 12 weeks of treatment with MB107, redness, swelling and desquamation at the lesions were significantly improved. Compared to baseline, it had a reduction of as much as 80% in PASI at a critical time, such as the trial endpoint, and a 50% decrease in PGA, indicating that MB107 has a potential in the treatment of psoriasis. The results of the second exploratory clinical trial in the treatment of atopic dermatitis, again, after 12 weeks of treatment with MB107, the redness and swelling at the lesions were significantly improved, and the EASI decreased by 69% and the IGA by 46% at the trial endpoint compared with baseline, indicating that MB107 has very good efficacy potential in indications such as atopic dermatitis. At the same time, MB107 is also a microecological drug, which has the function of changing the microbiota. It is now known that a key pathogen in atopic dermatitis is Staphylococcus aureus, and after the treatment of MB107, the proliferation of Staphylococcus aureus on the skin surface was inhibited, and such inhibition results are shown to be positively correlated with clinical efficacy. In summary, MB107 is a novel AHR activator that can be used to treat plaque psoriasis as well as atopic dermatitis. New drugs have been shown to be superior to tapinarof in vitro and in animal studies, and exploratory clinical trials on psoriasis and atopic dermatitis have also shown positive results. Based on the characteristics of our product, in addition to entering the market as a new drug, we will first apply for marketing in the form of medical devices in the short term to reduce the time to market. The second item is our new TCB nasal spray, which is new dosage form under development. The nasal spray dosage form of TCB is a multi-action microecological nasal spray product. The characteristics of this product are to regulate the bacterial composition and maintain the balance of the normal nasal microbiota, also to directly regulate immunity, and to maintain a healthy function of the normal host. It can also directly affect the structure of epithelial cells, adjust the function of the nasal mucosa, and achieve the effect of alleviating chronic sinusitis and allergic rhinitis. The market potential of TCB is currently explored in two indications. One is the treatment of chronic sinusitis and the other is allergic rhinitis therapy. The global market for chronic sinusitis was $2.13 billion in 2019 and is expected to climb at a CAGR of 7.4% to reach $3.5 billion in 2027. In the part of allergic rhinitis, the global market was $1.35 billion in 2020, and it is estimated that it will reach $18.1 billion in 2028 at a compound annual growth rate of 2.64%. Both applications are very marketable. The key mechanism in the pathogenesis of chronic sinusitis is the activation of Th2 responses, that is, the activation of excessive immunity. In vitro tests showed that the releasing of the two key cytokines, interleukin-5 and interleukin-13, can be effectively inhibited by the treatment of TCB, to the extent comparable with the effects of steroids. Besides that, we know that one of the characteristics of the TCB product is that it can regulate the bacteria in the nasal cavity, and we also identify the key good bacteria and the key pathogenic bacteria in the nasal cavity. We can also prove that while it inhibits the growth of nasal bad bacteria, our products can still promote the proliferation of nasal good bacteria. Thus, it regulates the bacterial composition of the entire nasal microenvironment, so that the balance of the nasal microbiota is not affected. Moreover, in the case of antibiotic destruction, TCB still has the effect of protecting good bacteria, so it can be combined with existing chronic sinusitis treatments, antibiotics, and steroids. We know that chronic sinusitis will actually recur repeatedly. The need for antibiotic treatment is actually because of the characteristics of the biofilm produced by bad bacteria. Biofilm will affect the inhibitory effects of the existing antibiotics on these bad bacteria. TCB is further characterized by the fact that in addition to being able to effectively inhibit bacteria, it can also prevent the development of biofilm to achieve the effect of avoiding the repeated recurrence of chronic sinusitis. In summary, TCB is a dual immune and microbiota modulator, which can be used for the treatment of chronic sinusitis and has the potential to treat allergic rhinitis. It has been proved in vitro to inhibit the function of Th2 immunity comparable to steroids, and also proved to have the function of inhibiting the growth of opportunistic bad bacteria, and has the ability to prevent the formation of biofilms, and can also downregulate the chronic inflammatory response caused by bacterial infection. We have completed some preclinical in vitro studies of such product this year, and we expect to conduct exploratory clinical trials next year to verify the efficacy in chronic sinusitis. Because the characteristics of this product are in the dosage form of nasal spray, we will also apply as medical devices first in a strategic way to shorten the timeline for marketing. The third is TB90010, a new microecological drug for the treatment of type 2 diabetes. This is an oral dosage form of microecological drug. TB90010 is the microecological therapy of type 2 diabetes. In fact, we have confirmed in vitro experiments and animal experiments that it has the effects of treating diabetes. This year, the exploratory clinical trial of MBS fermentation broth, TB90010, combined with metformin have been completed. Next, we are reporting the results of this exploratory clinical trial. The patients admitted in this clinical trial were still ineffective in the control of glycated hemoglobin HbA1c after treatment with metformin in such patients after receiving the combined use of MBS fermentation broth. 75% of them can maintain or reduce the HbA1c indicator, and increased beneficial bacteria and inhibited bad bacteria were observed in such patients. In the analysis of subpopulations, it was found that 50% of patients who were originally ineffective in responding to metformin could effectively reduce their HbA1c, that is, the levels of glycated hemoglobin were decreased by 0.58% after receiving simultaneous treatment with MBS fermentation broth, indicating that MBS fermentation broth has the potential to reduce glycated hemoglobin. Among them, in fact, we also hope to find out what kind of biomarkers that shared by responders, so that the MBS fermentation broth has such a function. We know that MBS fermentation broth is characterized by the ability to regulate the bacterial composition to improve the therapeutic efficacy of metformin, so we also analyzed whether there are biomarkers of specific gut bacteria in the population of respondents, which then make it beneficial for metformin therapy. We have also analyzed many biomarkers that respond to MBS fermentation broths and will be used to identify potential responders in the future. In the clinical trial of MBS fermentation broth and metformin combined treatment, as just mentioned, the subjects with type 2 diabetes who routinely use metformin and glycated hemoglobin still remain above 7%, participated in this clinical trial, that is, the subjects with metformin treatment failure additionally receive MBS fermentation broth to learn whether the efficacy can be improved. First, there was no significant change in the liver and kidney-related parameters, nor did there be any product-related adverse events, indicating that MBS fermentation broth has a very good safety and tolerability. In addition, in the analysis of gut microbiota, it was also found that the combined use of metformin can affect the key to diabetes, the increase of butyric acid-producing flora. In subpopulation analysis, glycated hemoglobin was reduced by 0.58% in the 50% of subjects who responded well. Among them, we also found important biomarkers of intestinal bacteria, which can be used as biomarkers in subsequent subjects to predict whether they can achieve stabilizing or lowering blood sugar levels after receiving TB90010 treatment. This is our progress in the development of new drugs related to MBS fermentation broth. In November 2022 this year, the United States approved the first microecological new drug, and also created the opportunity for microecological new drugs to be listed in the United States. There are several advances of this microecological new drug, including the breakthroughs of the existing regulations and scientific regulations, and there will be many exemptions in the implementation of human clinical trials, so that the listing of new drugs can actually exempt the PK test of the human body and related independent research. The United States has achieved such a market, and we also hope to follow such microecological new drug regulations to help our microecological new drugs be successfully launched in the United States. In the following, we will introduce the development of important subsidiaries of Microbio Group. The first important subsidiary is Microbio Shanghai, which was established in 2004 with an existing capital of 1.9 billion, and its main business project is technology development in the field of medical technology. Microbio Shanghai's goal is to become a heavyweight biotechnology new drug enterprise in China, and our strategy mainly includes these 5 items. The first is to accelerate the entry of existing new drug groups into the mainland market. The second is that we will combine our own technology platform to work with scientists on both sides of the strait to develop superior new drugs. The third is the M&A sales channel to build up commercial and revenue strength. The fourth is that we will establish a mechanism for the joint development of international new drugs through external authorization. The fifth item is to enter the capital market and build the strength of long-term operation. In fact, there are many advances in Microbio Shanghai this year, including a key breakthrough in nucleic acid new drugs, the development of new indications such as the COVID-19, which is currently being sent for IND this year to execute Phase 1 clinical trials in the United States and is expected to enter Phase 2 clinical trials in the second quarter of next year. The second new nucleic acid drug for liver cancer indications is also expected to be submitted for IND in Q1 in 2024. In addition, existing new drugs including ON101 are also actively applying for drug certificates in China, and then the FB825 antibody new drug in cooperation with Elio Pharma is also actively examined in clinical trials of asthma to verify the efficacy on asthma. The second important subsidiary reported is Cottonfield Organic, a chain channel for major organic commodities in Taiwan, established in 1994 with a capital of 500 million new Taiwan dollars. At present, the main business items include store operation, online e-commerce, delivery platform and organic farm, etc., and there are currently 68 stores in Taiwan, with the highest coverage rate in Taipei and New Taipei City. Cottonfield Organic has the following advantages in the market, and the first is the ability to independently develop exclusive projects. Cottonfield Organic cooperates with Microsoy and will independently develop healthcare products. The second is carefully selected international products. The third is to have the highest organic product standards in the industry, accounting for 100% organic in fresh food and up to 80% in containing frozen food or dry goods. In addition to high standards in the selection of products, we have also set up an inspection laboratory to conduct sampling and layer-by-layer checks on selected products, and we have also deeply implemented the concept of ESG. Thus, sustainable management and social responsibility are taken as the target policy of Cottonfield Organic in the company's management. The sales revenue of Cottonfield Organic has also continued to grow in recent years. The last item is in the part of biotech holdings, in addition to the existing subsidiaries of Microbio, including Cottonfield Organic Life and Microbio Shanghai. The holding areas of Microbio also include Microsoy and Oneness, Diamond Biofund, Sun Biofund, and Jingying Invest, and this year a new holding investment project is Microbio Singapore. Next is the valuation of the reinvestment, which uses the carrying amount of the equity method investment to assess its profit or loss. The original investment amount for the reinvested project was $3,963,066,000, which was $8,721,401,000 as of September 30, 2022. And the current investment interest recognized in the first 3 quarters of this year is $685 million. $6,000. This is an assessment of the return on our reinvestment. The goal of Microbio next year is to actively promote market access for all products in the development of new drugs and enter into the U.S. market. Whether it is intestinal ecology, in addition to new drug development, we will also use functional medical foods to quickly cut into the U.S. market. In addition, the application of medical devices for MB107 will be an important milestone next year. This is today's report on new drug research and development and reinvestment.
Thank you very much for participating in this session, and this concludes today's online meeting. In 2022, the company's overall operation is stable. Chemo Young oral solution revenue continues to grow positively, and the new drug research and development team has also continued to make breakthroughs and is planning to submit applications for U.S. medical foods and medical devices in 2023. We believe that in the new year, the team will bring more exciting good news. We hope that through this legal seminar, everyone can have a clearer understanding of the current operation situation and future prospects of Microbio, and wish you a happy new year and successful investment. Thank you.